Welcome to Unit 5 Lecture Video 2. So remember, Unit 5 is broken down into a whole lot of chapters. We're in the first chapter, Chapter 10. So in this video, we're going to talk about DNA replication. So DNA, deoxyribonucleic acid, replicating, it's copying itself. A couple of things about DNA replication. DNA is known as anti-parallel. So that means the strands are going in opposite directions. So if you look here, we've got the 5' to 3' end, and then the 5' to the 3' end. So that's what it means in the fact that it's anti-parallel. The 5' end means there's a phosphorus. The 3' end, means there's a hydroxyl group available. And that hydroxyl group, if you remember talking way back when at the beginning of the year when we discussed dehydration synthesis, that 3' OH end is really important for dehydration synthesis for adding in the next nucleotide. So remember our base pairing rules, A to T, G to C. We are not dealing with RNA at any point in this process, so it is A and T, G and C. There is no uracil. There are three main enzymes that we're going to talk about involved. DNA polymerase. Now there are multiple polymerases, but we're only going to just talk about them as a whole. DNA ligase, DNA helicase. Okay, and then because of this 3' to where we have to add the next nucleotide, there are two types of strands when we're building a DNA molecule. There's the leading strand and the lagging strand.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
So DNA replication steps, same, it's the same basic steps. DNA helicase opens up the DNA strand, creating that bubble. DNA polymerase is used to add the new base. So polymerase, we are polymerizing. The leading strand is synthesized continuously in the 5' to 3' direction. The lagging strand is synthesized in pieces called Okazaki fragments. So that's where they're organized in chunks. And then DNA ligase stitches those Okazaki fragments together, okay, to make one long continuous strand. And then this continues through the entire strand of DNA until there are two complete double-stranded molecules. Of those complete double-stranded molecules, one is the original strand and one is the new strand. So you can see here, so we start with the strand together, we open the, so we've got the strand together, we separate them, and then we fill in with the new bases, and then at the end we have two strands, one that are old, quote unquote, the template strand, and one that is new. So for your wrap up, I want you to fill in the table with these important words or phrases, and I will see you in class. Bye.